timely discussion here, timely lab here. We, earlier in lab, were talking about the sequence from superficial to deep, what structures are encountered with and surrounding the testes. And I remember talking about how each of the testes has this connective tissue structure that's called the tunica vaginalis, that is a serous membrane, so a little bit of fibrous tissue, but areolar, and of course squamous cells to make that serous fluid. And that tunica vaginalis is going to cover the testicle and the tunica albuginea. And that cremaster muscle was outside of all of that. And we talked about the cremaster muscle, remember? That the cremaster muscle and dartos muscle are both important for that temperature control, along with the pampiniform complex, the network of veins and arteries. All right, so we have the tunica albuginea is intimate to the surface of the testicle, and deep to it then are your lobules, and within the lobules are the seminiferous tubules. And we looked at those this morning, and now you know all the components of those seminiferous tubules, and the fact that they are separated by little strands of our tunica albuginea that continue on up to separate the lobules. And we know that the seminiferous tubules come in like packs of 4 or 5 within the lobule, and that they're really tortuous. And every one that you looked at this morning was perhaps the same tubule or a couple of different tubules, because we don't know. We just had good cross sections to look at all the cells within. The seminiferous tubules, of course, where we produce the sperm. And then in between the seminiferous tubules, we have the interstitial cells, and those interstitial cells are going to be producing the testosterone, the androgens of the male's reproductive system. And so, nice to see a familiar structure now, because you've had a chance to look at them microscopically. And remember that we talked about how the spermatogonia are the stem cells that are responsible for producing the sperm. The spermatogonia are going to be found along the periphery of the tubule, and they are then going to mitotically divide, and they're going to produce primary spermatocytes. And those primary spermatocytes are also diploid, as are the spermatogonia. What does diploid mean? Two sets of chromosomes. Where are those chromosomes? What are those chromosomes about? Where do they plug me in?
They're from your mom and dad.
They're from your mom and dad.
Millions of strands of DNA.
Strands of DNA. Yeah. 23 from mom, 23 from dad. Every single one of your cells in your body has the same sets of chromosomes. But the difference here is that these spermatogonium, that are stem cells, are going to mitotically divide and produce primary spermatocytes. And then these primary spermatocytes are going to actively get involved in meiosis. And meiosis is also known as reduction division. And it's called reduction division because the whole point of reducing the chromosomes is so that we don't put a sperm with 46 chromosomes together with an ova of 46 chromosomes. That would be crazy, right? And each time we would double those chromosomes. So we have to reduce the number of chromosomes so that when we put that sperm back together with the ova, then we get the right number of chromosomes again. And it's crazy how much that goes on. And look, you're walking, talking, working, functioning individuals. Yeah.
So when you have an extra chromosome or you have one less chromosome, is that because it didn't fully divide?
Yep, it's called non-disjunction. Yep. So, in between our seminiferous tubules, we have those interstitial cells, also known as Leydig cells. The spermatogonium are found along the periphery of our seminiferous tubule. And along with the spermatogonium, we also have cells called nurse cells, or sustentacular cells. Yeah, and they're very important because, as you can see, as I said to you earlier, they actually create that testes-blood barrier, and they surround all of these meiotically dividing cells. And remember that meiosis, the whole point of meiosis is to reduce the number of chromosomes, but also to mix up the genetic material. Because the more variability you have, the more variety you have, the better it is for the species. If we all had exactly the same DNA, we would all be toast by now because COVID would have taken all of us. If we have variety, then if we have variability in our DNA, then we have a chance to avoid some of those catastrophic diseases that can happen. So, there are people that survived the Black Plague. Those same people are avoiding HIV today. Isn't that crazy? You know what the Black Plague was? Oh yeah, in the 15th century, the 14th century, and the 15th century, there was a bacteria that was carried by rats. Of course, they had lots of rats. People would get bitten by it and develop this horrible disease that killed
Is it
lots.
bubonic plague?
The bubonic plague.
Yeah.
Yeah. Yep, killed a lot of people. But some people survived. Why did they survive? Because their genetic makeup provided them with that immune response. Yeah, and they're the ones that are actually avoiding HIV today. So people that have survived and didn't develop HIV, were exposed to it, didn't get it, down the line, they probably had
Ancestors.
family, right, that they came from. All right, so nerve cells are really important because they take care of those secondary spermatocytes. Those secondary spermatocytes are the result of meiosis 1, and they are completely different material now, because during meiosis 1, not only do you have this DNA, chromosome 1 from mom finding chromosome 1 from pop, finding each other, chromosome 2 finds chromosome 2, they all snuggle up with each other, they have condensed and made copies, and then they swap pieces of genetic material called crossing over. And then they go through meiosis 1, and the duplicated chromosomes, those sister chromatids, line up along the metaphase plate, and they could line up on either side of the metaphase plate. You might have mom's version on this side, dad's version on this side, dad's version, mom's version, dad, mom, dad, mom, down the line. It doesn't matter what side of the chromosome or the metaphase plate those duplicated chromosomes end up on. Anaphase 1 separates those sister chromatids from each other, from the duplicates. And now we have secondary spermatocytes with one or the other of those duplicated chromosomes in it. The secondary spermatocytes then have to undergo meiosis 2. But in the meanwhile, we have these nerve cells taking care of those secondary spermatocytes and making sure that they have coverage, that blood-testis barrier. They also secrete this protein called inhibin. So if the sperm count is really high, we don't need any more follicle-stimulating hormone to come from the pituitary gland, because we already have plenty. So the inhibin is a negative feedback saying we don't need as much follicle-stimulating hormone to make sperm at the moment because we got plenty. So these nerve cells are really, really valuable. This is a great example of how these nerve cells wrap themselves around our developing spermatocytes. There is a problem with this particular figure. Can anybody figure out what that problem is? There's a mistake. Yeah, Kesa?
Isn't it the secondary spermatocyte, it should be diploid, not haploid? Or haploid, not diploid?
Exactly right. Exactly right. So here's our 2N, our diploid spermatogonium. Diploid spermatogonium undergoes mitosis, produces 2 new cells. One of them is going to stay back and become another spermatogonium. The other one's going to become a primary spermatocyte. This is also diploid, so it's got a full complement of chromosomes. And this is where all the magic happens during meiosis 1. Crossover, independent assortment, all of that genetic mix-up. This primary spermatocyte undergoes meiosis 1, produces 2 secondary spermatocytes. These are now haploid. They have one or the other version of that duplicated chromosome in them. Could be moms, could be dads, could be all moms, could be all. And this is why you look kind of like your uncle. Seriously, there are pieces of your family that show up in you because this happened to your father. This is how your father was created. This is how your mother was created. This is how their mother and father were created. This goes on and on and on. So we carry this genetic material with us. And just as an aside, and I don't know if you've lost family members. Sorry. But if you think about it, you're carrying their DNA. And so if you get kerflumped, just think about that. Okay, back to secondary spermatocytes. So these are one end, and these are then going to meiotically divide into spermatids. So each of them is going to, these secondary spermatocytes, they have duplicated chromosomes in them, sister chromatids. Those sisters are going to line up along the metaphase plate. Sisters get separated. Now we have 4 spermatids, and each of them has one chromosome. It's mom's version or dad's version, but it's also got a mix-up of genetic material. When we go through this meiotic division, we have the potential for 8 million different genetic combinations. And then when you put that sperm together with the oocyte, you have 64 billion possible genetic combinations to produce you. I think meiosis is just the most amazing thing. So we want to remember that we have spermatogonium and this whole meiotic division going on, but we also have these nurse cells. The Sertoli cells are all known as sustentacular cells and nurse cells. And see how this nurse cell's cytoplasm is creating a barrier, and these cytoplasms are wrapped around these dividing spermatocytes, protecting them, keeping them from any potential antibodies that would recognize them as foreign antibodies. So again, we have our spermatogonium. They are 2N. They undergo mitosis. They produce 2 daughter cells. One of those daughter cells remains a spermatogonium. The other becomes a primary spermatocyte. This primary spermatocyte undergoes meiosis 1, where we have crossover, independent assortment. And that primary spermatocyte produces 2 secondary spermatocytes, each of them with a completely different genetic combination in them now. But these are N, yes, they're N, but they still have duplicated chromosomes in them, sister chromatids. And then they undergo meiosis 2, and meiosis 2 produces for each of them 2 spermatids. Those spermatids ultimately become known as spermatozoa. You can see this connected spermatids here. This is the leftover cytoplasm of the nurse cell that was wrapped around it. And so males are producing sperm every day, all day long, lots of them being produced. They are known as gametes when they're finally released, and they are released from the wall of the seminiferous tubule into the lumen of the seminiferous tubule, and the fluid that's contained in that lumen is produced by the nurse cells. And that fluid is going to carry those sperm to the reticestes. You were looking for the reticestes this morning. That's the channels that all the sperm are delivered to, that then go through the efferent ductules directly to the epididymis and stored in the epididymis. And if they're not used, they're resorbed, they're recycled, all the pieces. So here's our spermatid, and that spermatid, remember, is just a little cell with a dark nucleus, and this one looks like it's developing a tail already, and it's developing an acrosome. This acrosome is important. It's a packet of enzymes that's going to be used to break through the zona pellucida, the corona radiata and zona pellucida of the oocyte. And then we got a little bit more mitochondria developing here. Those mitochondria are going to be super important because without the energy, this is a non-modal structure. But the mitochondria is going to receive nutrients from seminal fluid. And once the mitochondria are exposed to the seminal fluid and you've got fructose, then these spermatids can start making energy. And then they can start driving their tail. So the acrosome is developing here, the tail is developing here. We start to develop a midpiece. Here's a head, there's the acrosome, nice cap on the head of the sperm. Our midpiece, all of our mitochondria are lining up, and then our tail. Ultimately, here's our head with the acrosome, it's just a packet of enzymes. There's the mid piece with all the mitochondria, and then the tail. And then they can swim. Spermiogenesis is what that's called. Making the sperm. As you see, it takes several days to get this done. But again, it's happening on a continual basis. The nerve cells stay behind, but the spermiation process occurs, look at that, 400 million produced per day. That's a lot. Well, when you see what happens to them along the way, once they've been released and they've delivered to the vagina, it's pretty hard. Harsh environment. Not a sperm-friendly environment. The sperm, well, they have been developed in the testes and stored in the epididymis. When they're called upon, when there is intercourse and the need to move those sperm, they will go from the epididymis down to the tail of the epididymis to the ductus deferens or the vas deferens. Remember, they're non-motile while they're stored in the epididymis. And that motility comes from the exposure to seminal fluid from the seminal vesicles. I want to show you this. So we used to think that sperm had a certain way of swimming. But we've discovered not only that sperm are motile, but that they can get to where they're going. And it's also very important that they are
In this
made
video,
correctly.
we'll be looking at sperm specimens with varying motility scores utilizing the microscopes in our fertility laboratory. As a specimen with more and more magnification, we can see that the sperm motility is pretty good. Most of the sperm are progressively motile. Sperm motility scores represent the percentage of sperm that are actively swimming in a forward direction. Low sperm motility is a common cause of infertility. This specimen shows both a good count and a good motility. Here it is at higher magnification, where we can get a better idea of the morphology or shape of the sperm cells. This specimen demonstrates both a low count and a low motility. There are many immature sperm forms present, and the progressive motility is poor. Here is another example showing a poor count and poor motility. This one also shows significantly abnormal morphology. Here's one more example of sperm with a very low count and a poor motility. We see that the sperm are twitching and wiggling in place, but there is no progressive motility. In order to get a pregnancy with sperm like this, we will need to perform IVF with ICSI and inject the sperm directly into the eggs.
Wow, crazy. That, and then there's just one more, and this one is kind of interesting because it shows you the, all right, never mind. I didn't check to see if it was still available. But I'll find another one for us. Sperm have to be able to move forward. They have to be able to swim in a progressive direction. They also have to be made correctly. They can't have two heads. They can't have two tails. They can't have too long a tail, or too short a tail, or a bent tail, or a bent head. It doesn't seem like there could be that many problems with sperm because they're so simple. But no, they have to have just the right amount of everything in order to be successful. And even if they're made perfectly, again, the environment, once they leave the urethra, not It's hard on them. So, that's the sperm story. Okay, that's the end of that show. Let's start part 2. So, you all remember puberty? Wasn't it fun? Yeah. Obviously, you're young, you don't care, you're growing, you're having fun, and then somewhere around the age of 12, things start to change. You start to feel a little different. Well, that's referred to as puberty. And puberty is the result of this surge of, as this says, gonadotropins from the hypothalamus. Gonadotropins from the hypothalamus. What do they stimulate? They stimulate follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone is important for both males and females because it's going to be producing sperm. We need follicle-stimulating hormone to make sperm. Well, males and females hit puberty a little bit differently. Males are a little later when they hit puberty than females. And there's all kinds of reasons for that and rationales for it. And they think so when we were back in the caves, there was a division of labor. And it would be required that the male go out and do the hunting and the female go and do the gathering and the taking care of the babies. We would need a little more mature male to be able to impregnate the female. And so that was part of what they think is this evolutionary process, reason why males develop hypothalamus-wise a little bit later than females do. You can read this article from the National Institute of Health if you're interested in the evo-devo of human adolescence. But puberty is, as it says, the onset of those gonadotropins, follicle-stimulating hormone in particular for both males and females. Follicle-stimulating hormone getting the ovary up and running, and then for males starting the production of sperm. Yep, see, it's usually, finishing up puberty at 12, 13, 14 in boys. That leads to a period known as adolescence, where all your hormones are in a complete and utter uproar. Yeah, puberty. So, we have the neonate, we have the child, we have the adolescent, we have the teenager, and we have the adult. And this same thing with the baby and the newborn, the 1-year-old, the adolescent, the teenager, adolescence, and then maturity. And you can see here it's within this range that we have our puberty. And what is puberty all about? Well, again, it's the release of those gonadotropin hormones from the hypothalamus that stimulate the pituitary gland to release follicle-stimulating hormone and luteinizing hormone. The follicle-stimulating hormone responsible for producing sperm in males, luteinizing hormone responsible for stimulating the interstitial cells to make testosterone. Were you all able to find some interstitial cells this morning on your slides? Yeah, good, okay. So it's this important production of testosterone that's going to drive the sexual maturity of that individual. So we need those follicle-stimulating hormones luteinizing hormones to produce testosterone that is going to drive the sexual maturity of the individual. And then follicle-stimulating hormone to drive the production of sperm. And that secondary sex characteristics, you might remember, was more muscle, right? What else happens with the secondary sex characteristics of males?
Hair on the height.
Did you say something? What did you say?
Larger skeletal structure.
Larger skeletal structure.
Facial hair?
Facial hair.
Deeper voice.
Deeper voice.
Laryngeal prominence.
The laryngeal prominence. Right, all those characteristics that provide for the selection process. Right, the selection process which is done typically by females. Yeah, so we have the testosterone that's driving that, then certainly as the result of the onset of puberty and those secondary sex characteristics. And big growth spurts, that's for sure. And so Here we have our gonadotropin-releasing hormone from the hypothalamus, stimulating the pituitary gland to produce follicle-stimulating hormone and luteinizing hormone. And follicle-stimulating hormone is important for the release of androgen-binding protein from the nurse cells. So we have something that is, what was that called? Synergy in endocrine system, where we have two hormones that are responsible for doing the same thing or getting two parts of the same thing together, right? Synergy, yep. So we need follicle-stimulating hormone and the nurse cells to produce androgen-binding protein to be able to stimulate this division of the spermatogonia to produce the sperm, resulting in spermatogenesis. No effective testosterone without follicle-stimulating androgen-binding protein. We still need the testosterone. The testosterone is going to participate in this whole process as well. But without the follicle-stimulating hormone and the androgen-binding protein, testosterone won't have any effect at all. So that would be the permissive part of that whole process from the endocrine system. Follicle-stimulating hormone plus the androgen-binding protein released by the testicular cells to stimulate the spermatogenesis, along with the presence of testosterone that allows for the production of sperm. And that negative feedback system, right? If there is, as it says, plenty of testosterone, then we're going to have inhibin released, and it's going to prevent more follicle-stimulating hormone, more gonadotropin-releasing hormone. It's going to prevent all of that from being released, which then is going to mean that we reduce the number of sperm that are being produced through the reduction in testosterone. Can everybody see that? Right? So we need this and this and this and this all to do to make this. It all has to work in conjunction, right? We have to inhibit the gonadotropin-releasing hormone, which then means that we're not going to have the follicle-stimulating hormone and the luteinizing hormone, which means we're not going to have any of that happening if there's plenty of testosterone already. If there's plenty of sperm already, we're going to have inhibin released by the nurse cells. That inhibin is going to do the same thing, prevent any of this from happening up here. So a little negative feedback going on there. You all are familiar with the phrase menopause. Menopause is the cessation of menstruation. Well, males experience a similar, but not quite as dramatic, an event. It's called andropause. And it really is not as hard and fast. Like their testosterone levels drop, but they don't stop producing sperm. Males continue to produce sperm, maybe not at the same level. The testosterone, maybe not at the same level. But look, it can actually start pretty early on, right? Even in their 40s and 50s. You see even lowering of testosterone in males in their 20s. This all could contribute to lower sperm counts, and it could affect libido, which is the drive, sexual drive. And it may impact the ability for somebody to maintain the erection. Yeah?
Are there specific factors that lower testosterone? Like environmental factors or stress?
Probably, yeah. Yeah. Stress for sure, because that does it to everything. Yeah.
What is it that causes an enlarged prostate? Why does that typically happen?
Yeah. That's because of the presence of testosterone. Yeah, the benign prostatic hyperplasia. So I was dating a veterinarian for a while, and his whole thing was that if we castrated male humans at the age of 40, we'd have a reduction in prostatic enlargement. Of course, it's not a very popular opinion.
Point of view there.
He really wasn't, talking as if he was planning on promoting it. He just was saying that it is testosterone that is the driver for enlarging the prostate. And if you want to stop the enlargement of the prostate, you got to get rid of the testosterone. And how do you get rid of the testosterone? You got to take the testes. Because a vasectomy is going to stop production of testosterone. That just stops the delivery of sperm. Yeah. So yeah, it's related to testosterone levels. Yeah. There's ways of dealing with that. Yeah. Yeah, so one of the things that does happen, is that the prostate can enlarge as a male gets older, and that enlarged prostate can impede the ability to empty the bladder completely. And that's why men in their 60s and 70s might be finding themselves visiting the bathroom several times at night because they're always maintaining a semi-full bladder. They can't empty it because the prostate wraps around the urethra. And when it gets bigger, it squeezes the urethra. And that makes it more difficult to urinate. And developing the erection as well, because you have blood supplies heading down that direction as well. And so, we've now talked about the testes. We know about the production of sperm in the testes. We know that that sperm is then delivered via these efferent ductules to the epididymis. And the epididymis is considered part of the duct system for the male reproductive system. The epididymis leads to the vas deferens or the ductus deferens. The ductus deferens joins with the seminal vesicle and delivers directly into the prostatic urethra via the ejaculatory duct, the fluid that is delivered by the seminal vesicles and then down through the urethra. So the epididymis is the first thing to talk about. And it is, as I said earlier in lab, the storage unit for the sperm that's produced in the testes. The epididymis, I want you to note, is just cupping around the surface of the testicle. It ends at the bottom here. This is all ductus deferens. So we have a head, a body, and a tail of the epididymis, and then the ductus deferens all the way up. And see how it loops over the ureter? That's something that we couldn't really see when we were looking at the models earlier today. I want you to note that there's a loop up over that ureter. And the ureter, of course, is delivering urine to the bladder. But notice that the prostate gland is surrounding the urethra. And if it gets bigger, then it can occlude the urethra, which makes it very difficult to empty the bladder over time. We identified the epithelium of the epididymis, pseudofibrillate ciliated columnar with the stereocilia, especially long cilia that keep moving the sperm along. Again, they are stored in the epididymis where they get to mature a little bit. At this point, they are non-modal. They don't have the fuel to make the energy to drive the tail. So they're just waiting there to be called upon. How do they get from there uphill?
Peristalsis, smooth muscle.
Peristalsis. So smooth muscle surrounding our vas deferens, delivering that sperm to the ampulla of the vas deferens, which is right here. And then, as I said, the ampulla joins with the seminal vesicle or seminal gland to form the ejaculatory duct. Yep, so I'm kind of surprised that we had a discussion about penile implants, was really fascinating because some men, suffer from erectile dysfunction for a host of reasons. So good that they've created a mechanism by which they can sort of gain a little bit of a sense of life again. Another mechanism by which we can manage the male reproductive tract is through a vasectomy. The vasectomy is to sever the vas deferens or the ductus deferens so that even though sperm is produced, it cannot be delivered. So it's a pretty efficient mechanism for birth control. It's not one that you go into lightly. If you want to have babies in the future, it is not recommended that you get a vasectomy, even though they say they can be reversed. there's no guarantee that once this has been cut and separated, that you can put it back together and still have a patent tube. Because you're gonna probably have some scar tissue. Yeah,
So when they just snip it, they just separate it in what? The sperm that was produced by the testes that one's gonna go through the ductus deferens just is reabsorbed?
yep. Yeah, so if we don't use the sperm, they're reabsorbed all and recycled. Yeah, so that happens on a regular basis if you have a testicle that's not being called upon to make sperm every day or every week or whatever. They don't back up. No dead sperm backup.
But seminal fluid will still be released.
Yes.
Yeah.
Yep.
So
Because
where
the
does
seminal
it?
fluid is released from here.
Okay. And so it's usually cut like down further.
I'm not sure exactly where on
they
the
cut it.
ductus deferens they go.
I mean, there's plenty.
It's through the
Plenty of
spermatic
opportunity
cord,
to
so it's
cut
easy
it.
for them to find it. And,
Right.
yeah, pretty simple procedure. And they would ligate, and then take a chunk out. That way, there's no chance of it rejoining on its own, potentially. Yeah.
I have a quick question.
Yeah.
If you cut the tube and like the sperm just stays in it, does that like affect you in any way, like not releasing sperm?
No, they just get resorbed.
Okay.
So don't let anybody tell you. Yeah, so what else about this? The ampullas, we remember how the seminal vesicle or gland and the ampulla of the ductus deferens merge to form this ejaculatory duct, which then travels through the prostate. These are the next things that we'll talk about, are the glands. But once the sperm joins the seminal vesicle produces a really rich fluid. It is the fluid that bathes the sperm and provides them with their first nutrients in the form of fructose. And they have all the mitochondria in their midpiece, and so they can go through cellular respiration to make ATP. And their whole job is to drive that tail to get those sperm moving forward. Yeah. And then that fluid from the seminal vesicle carries the sperm through the prostate gland. The prostate gland produces its own fluid. So now we have seminal fluid and prostatic fluid. We can now essentially call it semen, because we got sperm and we got seminal fluid and prostatic fluid down the length of the urethra. And we have little openings right here for the bulbourethral gland. And the bulbourethral gland, right here, that bulbourethral gland produces a lubricating-like fluid. And it's actually the first fluid to be released. So this bulbourethral gland is going to release its fluid, and that travels down the urethra to the tip of the penis, and it flushes out any potential urine that would be acidic, that could be detrimental to the health and well-being of the sperm. So we flush the urethra out, and then we deliver our sperm down the length of the spongy urethra and out via the external urethral orifice. That seminal fluid, really important, because as I said, it provides fructose for getting those sperm some energy. You've heard of prostaglandins before. These prostaglandins are ubiquitous, they're all over the body, and in this case their job is to get that smooth muscle contracting to drive that sperm up the ductus deferens and then down the tube. Get it out. Proseminogelin is really cool. Look, it's activated by seminogelin. You got this production of fibrinogen in the semen. It makes a little plug so that the sperm that's delivered to the vagina now has the ability to create a little plug so it doesn't drain out too easily. Because once it's delivered to the vagina, you want it to get up through the vagina. You want those sperm to have a chance. So the proseminogelin comes from the seminal vesicles, the seminal glands. And look at the vast majority of the fluid that's produced in this system comes from those two glands. Yeah, nice little clot in the vagina to hold that fluid in place. You got a little calcium in there. Why is calcium valuable? You remember calcium and how important it is for skeletal muscle contraction, for nerve innervation. Now it is a little driver for keeping those sperm going. The next of the glands is the prostate. And as we saw, the prostate is a single gland wrapped around the urethra. It produces more fluid. And I don't think it necessarily is seminal fluid. I think of seminal fluid as from the seminal gland. This is prostatic fluid, but collectively, semen is now being produced. It contains citrates. They're not quite sure what the role of those citrates are. Maybe some nutrients for sperm. Hard to know. Definitely got something to do with the mitochondria, maybe to produce more ATP. You got your more proseminogelin converted to seminogelin. Cool. This is going to liquefy that sperm clot after it's been in place for a while. So you don't want it to stay there indefinitely. After a while, it can either make it up or not. Seminal plasmin to prevent UTIs maybe in males, so another mechanism for flushing out the urethra on a regular basis. And then also some calcium is produced by the prostate gland, but here's the weird part about it. In this case, they think the calcium slows the sperm down. And what do you think the value of that would be? Why would you want to slow the sperm down?
So you don't get pregnant?
to keep them from spreading themselves like out too much. I don't know.
So you can have sperm delivered to the vagina and get up to the oviducts a couple days before ovulation.
Yeah.
And if some of those sperm are held back, they have a chance to fertilize that oocyte several days later. So that's the reason for having the slowing of some of the sperm, so that they're held in abeyance. There's a whole lot of things that can happen to these sperm. They get hung up in the walls of the uterus. It's not perfectly smooth. They get hung up at the cervix. There's a plug that the cervix produces. There's all kinds of things that can impede their movement. But if everything is flowing, then you might want to make sure some of them don't get delivered too soon, because then that's kind of wasted the sperm. How many a day? 400 million? Okay. Semen, a little bit on the basic side, right? A little bit on the basic side. Those glands, so here's our seminal gland, here's our prostate, here's the bulbourethral. Bulbourethral delivers that fluid first, flushes out the urethra, provides a little bit of lubrication for the tip of the penis. The seminal vesicle and the prostate make their fluid that's delivered via the ejaculatory duct and the prostatic urethra to the spongy urethra. And then next to talk about is the penis. Remember, it is one of the two external genital structures, scrotum and penis, two external genital structures. It is a it is a blood-filled structure, yes. Not always filled with blood, but space for blood, and obviously a good blood supply for the penis. The penis has the root, which we're not showing here on this image. There's a root. You did see it in the little models. Remember the 1/5 life size? The penis had the two little, right, those are called the crus of the penis, and then the root is just where they meet. That's the root of the penis where it's attached to the body wall. You have the shaft of the penis. This is the shaft of the penis. You have the glands of the penis. And the glands of the penis is covered with a skin known as the prepuce. This image right here is showing you the penis with the prepuce intact. This image is showing you the penis with the prepuce pulled back. And the prepuce is pulled back because it's been cut and it retracts and it exposes the glans penis. Here they're showing you the prepuce has been pulled back a little bit. In an uncircumcised male, this is what a relaxed penis looks like. In a circumcised male, this is what a relaxed penis looks like. We were talking about that earlier. Culturally, circumcision is a big deal in this country, not so much in necessarily the rest of the world. I understand there's a movement to cease circumcision. It's really a personal decision. They originally thought that males couldn't be held responsible for keeping their own penis clean. And so they needed help. That's why they did it. Yeah. Circumcision. I have a really interesting little video on circumcision of the male penis. Cross-section of the penis, all the blood sinuses. There's our deep arteries. There's our superficial dorsal veins. These are our corpora cavernosa. There's our corpus spongiosum and our urethra. There is a septum that's dividing the two corpora cavernosa. You have, of course, fascia around the outside. These blood sinuses, or whatever we're calling them on this slide, blood sinuses, blood cavities, venous cavities, this is where we fill the penis up with blood, and that produces the erection. So I want to show you what happens. So here we have the shaft of the penis, and we have our deep arteries here within our corpora cavernosa, and they have, from the deep artery, branches of the Helcine arteries, and the Helcine arteries provide the blood to fill these blood sinuses, or sinusoidal spaces, or venous spaces. And when blood fills those spaces, it actually presses on the venous return vessels. And so as long as there's stimulus to produce the drive of blood into these venous spaces, these blood sinuses, that blood cannot drain out of the veins because they are pressed closed. That's how you sort of maintain the erection, is by pressing closed those vessels. So these guys deliver blood to all these blood sinuses via the Helcine arteries. And then the Helcine arteries fill those blood spaces. And it's nitric oxide that actually drives that process, because the nitric oxide relaxes the capillary spaces, those spaces. When we have our penis getting involved in the process of reproduction, we know that there are basically 4 phases for that process. The first phase is excitement. Masters and Johnson, have you heard of them? Masters and Johnsons were the 2 researchers that finally came up with all of this. They researched hundreds and hundreds of couples.
Did they make a movie about
They
that
did.
with Liam Neeson?
Yeah,
Yeah.
yep, yeah. I think it's HBO.
Is it? I don't know.
I think it is.
I
Yeah,
saw
so
it.
there's a movie on it, how these 2 individuals, a man and a woman, Masters and Johnson, came up with this whole process by which they observed many, many, many couples having intercourse. And they honed it down to this. We have 4 phases for sexual intercourse.
Someone's got to do it.
Hmm?
I said someone's got to do that research, right?
Somebody had to do it, right? So first phase is excitement. We're familiar, probably. The second phase is the plateau, where everything is enlarged, ready to go. The third phase is called the orgasm, which is divided into emission and expulsion. Emission is when all those glands are producing the fluid, and the sperm are being dragged from the epididymis up through the vas deferens and into the urethra. Right? Now they're bathed in the fluid. Now they've got some energy, they can swim. Expulsion is ejaculation, and then resolution. Deflated penis.
Flip the pump.
Flip the pump. Yep, that's right. So where does that all blood come from? So your iliac arteries. Do you remember them? Abdominal, right down here? Mm-hmm. So all that blood is traveling to your penis via those arteries, those dorsal arteries, or excuse me, the deep arteries, also from the dorsal arteries. Yeah. And remember me telling you that this is the only time in your entire body where your sympathetic and your parasympathetic system are working in conjunction with each other. As you can see here, parasympathetic releases nitric oxide. What does that do? Oh, yes. Increasing the size of those arteries, increasing the space of those lacunae, as they call them, right? Because this is going to fill those spaces up with blood. So the parasympathetic innervation. Sympathetic innervation, where we get more dilation of the blood vessels and more erection. And also getting some sexual thoughts going on there too. By the way, the glans penis is loaded with so many sensory receptors, like it exceeds anywhere else in your whole body. The glans penis, and you can understand why. Because that's where it all starts. Excited, right, exactly. So we have here our excitement phase. Where does excitement come from? There's always the mental, of course, and then there is the stimulus. And you have the mental and the stimulus together, and you end up with an erection. Then we have, from the erection, our emission phase, where all of those fluids are getting all wound up and delivered. And then we have the ejaculation phase, right? More sympathetic innervation here. And then finally, resolution. Once you've ejaculated, there's no need for any of the stimulus to go on, and so you get resolution and deflated penis. And that, my friends, is the end of the male reproductive system.
So how does the refractory period factor into that?
Pardon me?
The refractory period where you can't get erect again for a short amount of time?
Oh, yeah.
After you orgasm?
It probably depends on the individual. Imagine that's like partly an individual.
A skill issue?
How do you want it?